So we can start.
Okay. Okay. So, okay, welcome again, all of you. And today is the lecture 4 for the synthetic biology and the space bioengineering, and is the last lecture for this particular subfield in this course. So, in my last class, actually showed this as my last slide, where I talked about particularly Professor Richard Feynman's comment, and it is nothing to do with the biology, but what he very importantly put, that what I cannot create, I do not understand. And till now, when we studied the biology, if you think about studying from our old days of the biology, like the botany, zoology, I mean, when the idea of animal, idea of the plants and their systematic studies have been started long back, several hundred years back. That time, from that time to almost like end of the last century, what we have actually gathered a lot of information on the biology. And at this present moment, the amount of the data we are getting in unprecedented scales, right? However, as we also explained in our first class, that we know a lot of information of the biological system, but we do not understand actually what is happening all that time. And that is nowadays people think that's getting so much data is actually a challenge, because we do not know how to analyze those data and try to get some proper fundamental laws of the biology, and then which law you can use to develop something else. Now, this is so one way to, as I mean, when we actually think about, you want to build a rocket, right? And this rocket goes through it follows a particular principle, like the Newtonian mechanics. So this, by building, you can understand that whatever we know for just a mathematical equation, a lot of ideas about even the Newtonian mechanics actually developed. Not the developed, but its applications you can find in the mechanical engineering and all those things. So one idea to understand a system is by building things, and that is what Professor Feynman actually pointed out. Now, today, we have some way of the, I mean, synthetic biology is just a start, but we have some way that we can actually start building biology, or start building biological system. So one way, so till now what we have tried to understand from a lot of biological data information we gathered, and tried to tell, okay, now we explain that this biological phenomena works in that way, right? So, but now we have the tools that so that we can actually start building the biology based on the whatever the laws or the rules we think about the biology. So when we start building these things, our understanding of the whole system would be much more deeper, and the synthetic biology can give a direction or a tool or an approach actually to do that. So here we explain one example only, and in little detail, that's how we can understand biological system by building. So let's take an example, and which is called the biological switch. So we can see this biological switch is almost everywhere. So now think about the human developmental biology. So we have started our life from a single cell, and now when you have an adult human, so there is a billions of the cells or trillions of the cells. Those cells have basically have the same genetic information. I mean, which has a nucleus, and inside nucleus which has a chromosome. That's definitely the red blood cells do not have a nucleus. It doesn't have any genetic information. But the other cells which has this nucleus and this genetic information have the exact the same genetic information than the other. Yet, what we find that every cells, I mean, from a different organs works in a differently. It means like the kidney cell works differently than a heart cell. or a skin cell works differently than lung cells. So this is when it is happening during the development that some cell is going to be make your lung, some cell is going to make the liver, some cell is going to make the kidney, some cell is going to make the heart. So that means that there is a bifurcation in that terms, that one cell is just going do the function, and what its whole genetic network will actually support that the function whatever the kidney cells is performing, and another cell which is going to be heart, its genetic network is turned on and off in such a way it will only support the function of the heart, or that's why you can call it as a heart cell. Now, interestingly, what we see in an adult human, it never happens that suddenly our skin cell started making a kidney cell, or our kidney cell started making heart cells, right? So if it is going to happen that a heart cell spontaneously start making some kidney cell, then as a human we'll die. The whole human life, or the Homo sapiens, which is for several thousands of years, it won't survive. This is it's not just the human, but any other animal, the plants, it cannot survive. So which basically means during the development, when this bifurcation happens, there is a switch, and the switch actually tells that actually which set of the genetic network should turn on to make a kidney cell, and which set of genes actually have to be turned on in order to be in a heart cell. And this switch, as I mean, in the first class, I tried to also explain, a switch is a simple memory device. So this here, there's a lot of the switch. For a long time, we knew that this kind of developmental switch is totally irreversible. You cannot bring them back. But now we do know that you can actually bring a skin cell to pluripotent stem cell, and that from the stem cell you can actually, I mean, induce stem cell to pluripotent stem cell, and then stem cell you can actually go towards an heart cell or something. So it is exact. So this, I mean, there is a debate that even you can call this as a reversible switch or not. But what I'm trying to tell you that now we know that it can be switched from one cell to another cell, and another cell to again you can bring it back to another cell, because now we have the techniques. But in what happened, that means though it is can be done, but in reality these switch are very robust switch. That's why a kidney cell spontaneously cannot change to a heart cell, or a heart cell cannot spontaneously change to a liver cell. So there is some switch somehow biology actually developed. And so this is what kind of this developmental switch I was talking. You can, I mean, understand that those are very complex switch. But now if you come to a prokaryotic system, just a bacteria, and then you can see there is also biological switches are present. And what kind of the biological switch? So one of the example is that a lysis and lysogeny cycle of bacteria. Now, let's say E. coli is the bacteria, and there is a bacteriophage. That bacteriophage actually attacks this E. coli. And when it attacks this E. coli, it writes, first what it does, it writes its genome into the chromosome of the bacteria. Okay, and it stays dormant there and without doing any harm. And this is called basically the lysogeny state. And this can stay for years after years, even 100 years, without doing any harm to this E. coli system. Okay, and this is a lysogeny phase. Now, if there is a change in some of the biochemistry of the cell, for example, you give an UV radiation to that lysogeny state of the bacteria, then due to this biochemistry change, this lysogeny state now go towards something called lytic stage. And in this lytic stage, what this virus starts copying itself like crazy, and then makes lots of different viruses inside the cell, and break opens the cell, and those viruses actually come to the environment. So this is a lytic cycle. So this lysogeny and lytic cycle is also a switch, and we'll see in a moment that how it works, because this is a kind of the switch humans studied probably the best. I mean, that is the biological switch what human studied the most. We know almost everything from molecules to molecules, every mathematical description of it, and so on. So that is the biological switch. Now, what the question synthetic biology is going to ask, okay, so there's a lot of biological switch starting from this bacteria and it goes to the higher animal like the human, but how to build a biological switch? Okay, so we see that how synthetic biology is building a biological switch, and before that we have to know what is the engineering principle behind that switch. So now, as I said, the lysis and lysogeny actually studied very well. So there is a lot of, I mean, if you think the whole genetic networks, and now people know the whole genetic networks, and whole genetic networks actually contribute for this switch. However, there is, if you think what is the critical region of this switch, we can actually bring it down to the two proteins, and one is called CRO and another is called the C1. So when is the lysogeny state, means that virus is not doing harm to the bacteria. What happens, there is something, this C1, there is a lot of C1 protein actually present inside the cell. And this you can see here. So they actually produce a very interesting combination. There are two promoters. One is called PRM promoter, through which the C1 gene is going to be produced, and another is called the PR promoter, through which CRO gene is going to be produced. What is happening is that they actually share kind of a common operating sites. So when you have a C1, this C1 first binds with the OR1, that's the operating site there. Okay, and so this is, and then when it binds the OR1 site, due to some cooperative reactions, it actually attracts other C1, and that other C1 binds at the OR2, I mean, in this kind of the region. So now, which basically means if you have some C1 into your system, this CRO gene cannot get produced. And the CRO genes are responsible to start some other biochemical reaction, which will go to the lytic phase. So now, here RNA polymerase can bind and start producing the C1, and when a lot of C1 will going to produce this, I mean, if there's enough C1, this C1 going to again going to bind the OR3, because if you actually see what is the binding constant between the C1 and these three of them, you will see the binding constant between OR3 and the C1 is the least. And that's why at the very end, it will bind to the OR3, and even it is close the even the C1 production. So no, there's a lot of C1 are present. I mean, in terms of a steady state, there's a lot of C1 in the present in the cell, and this whole system has stopped and nothing is happening. Now, if you give the UV radiation, so there's some biochemistry changes, and what happens, these C1 proteins get chopped. Okay, so when it's actually decayed or chopped, then there is no C1 protein present. I mean, the C1 protein is reducing inside the cell. So now there is no C1 can be bind to the OR1 or OR2. At this moment, RNA polymerase, so it opens up the area, I mean, region for the RNA polymerase. Now, RNA polymerase can come and sit there and start producing the CRO gene, start producing the CRO protein. This CRO, what it will do, it will first bind with a very high affinity with the OR3. So when it is binds with the OR3, which means that RNA polymerase cannot bind here and it cannot produce the C1. So as I said, that if CRO start producing, then it can actually trigger some of the other biochemical reactions which goes to the lytic phase. So that whenever the CRO, it is not only turning on this lytic phase or lytic genes, it is also actually stopping the C1 gene. Similarly, we have seen that C1 gene, it is stops the CRO is the completely. So this is a molecular kind of the description. So when you talk about the switch, switch has some of the engineering property, and we'll come in my next slide that what that does that mean. So as I said, that people actually studied this whole system in a much detail in terms of the mathematics and something. So here we call something called C1 CRO null client. So this is basically nothing but in a nonlinear dynamic terms, this is a phase space. So what is there? I'll just explain it in a chalk talk, definitely. But here, first I want to show you if you want to take a real E. coli cell and actually measure the C1 concentration and the CRO, or number of C1 and number of CRO you actually measure, then you actually draw this phase space, what you'll find that, so the y-axis, I mean, so is this CRO and here's the C1. So now these two lines, so one is for CRO and another is for the CR1, they will cross each other in the three places. One is here, this red circle you can see, another is in this here, this is another red circle, and another is in here. So they will cross each other three times. And in these three times, it is this one, okay, in the top corner, it is a stable steady state. I'll come in a moment that what the means in the stable steady state, but just for the moment, just believe that this is a stable steady state, and I numbered it as a stable steady state 1. You could number it 2, it doesn't matter. And this one is the stable steady state 2. Okay, and now in the middle, this cross is nothing but an unstable steady state. Okay, so this particular phase space is called null clines, and it is very important to explain this switching behavior. And now I'll go in little details in the chalk talk that what basically it means. Okay. Now, so I'll transfer to this. So I hope you can see this paint, the white board, because last class I think there was something happens that we could not see this. So if you cannot see, then please let me know that you cannot see. Then I'll try something here that so you can see. So I'm assuming you are now seeing that. So So what this null clines is going to talk about, so it comes from a branch of the physics or a branch of mathematics, you can say, and that is the nonlinear dynamics. Okay. So I'll give you the example from the C1 and CRO, but you can put it as an X and Y or any other thing, any variable you can want to put it here. Okay. Now, nonlinear dynamics actually deals with, so any physical parameters or chemical parameters you are dealing under the nonlinear dynamics, it means that the differential equation is forming is nonlinear. So in nonlinear dynamics, if you have any parameter, that gives you nothing but a nonlinear differential equation. So what is, let's say I talk about one equation. Let's say it's dy1 dt, one equation. And if I write something like 2t, for example, 2t plus k. Okay. Now, if you see, so this part, if you think as a, it will take this as an y, and if you compare with it, you see this equation looks like a straight line, right? Straight line equation, right? It's a linear equation. So this is called linear differential equation. Now, if I write one equation which says that dy2 just and dt equal to 2t square plus k. Now, this is not linear anymore. It doesn't looks like y is equal to mx plus c, because here you have a square term here, or this equation could be sine t, for example. Okay, then none of this is basically a linear equation. So these are the nonlinear differential equation. So this nonlinear differential, it is very difficult to solve a set of nonlinear equation together. And I'm not going into the detail how to solve it, but just take for the moment, it is almost difficult. It is, I mean, when you have a 2 or 3 different nonlinear equation, is completely difficult to solve. So we can only solve those near the steady state. Okay, so that's something how we are going to do that. That is not the part of today's talk or today's lecture, but just to tell you that solving differential equation is a very difficult situation. Now, imagine that C1 and CRO null-clines. So I have two differential equation. One differential equation is, let's say, in terms of is the concentration of the C1, that's a variable with time, is equal to is a function of C1 concentration and CRO. I'm not giving the third bracket again, just writing as a variable, that's the CRO. So let's say this is one of our equation, right? And another equation is in terms of the CRO. So let's say D CRO dt is equal to F2 C1 CRO. So in both the cases, C1 and CRO are the definitely the variables, but the function is different. It could be 2 C1 square plus 3 CRO cube. We don't know. I mean, just I'm assuming. And then this could be 6 C1 plus 9 CRO cube or something like that. For example, like this could be happen. So this is the nonlinear two nonlinear equation. Okay. Now, at a steady state, so what is the steady state means? Steady states of any parameter which is not changing with time, which is here means at a steady state, this C1 won't change with time dt, and at the very same time the CRO won't change with the dt. That means this, we equate this with the 0. Okay, so at the steady state, so at steady state, then what I can write? I can write F1, that is C1 CRO, equal to F2. CRO at the steady state. So at the steady state, we can actually get, I mean, if you equate, then I can get two different kind of the equation, one in terms of probably in the C1, another in terms of the CRO. It is like 2x plus 3, so 2x plus y, you are combining with 3x plus something y or something, whatever. So we can actually, we are equating that. Now, what is null-clines? Null-clines is how these variables at this, it actually in a phase space, how instead of the time, if I just try to, let's say here our dc dt and here dc or dt, instead of dt, if I actually draw something which is C1 and CRO, okay, then we'll got, because in a nonlinear equation we got something nonlinear, it looks nonlinear. And what is the nonlinear? Let's say one curve would be something like that, okay, and another curve may look something like that. So now, where this is the steady state, now as I show here, that at the steady state, this function and this function is the same. So wherever they are crossing, right, these two equation, one is F1 and is F2, which basically, so they are the steady state, right? So this is one steady state, this is one steady state, and this is another steady state. This is the steady state. Actually, it is happening in the C1 and CRO. We have seen there are the three steady state, because where these two functions are the same. And when it are same, when this is a dCRO dt and dC1 dt is equal to 0. So if we look at this kind of the null clients, through the null clients, we can understand where is the steady state is. Now this steady state has some property. What is that property? And this property, is it, so the question is it a stable steady state? Or unstable steady state? Okay. Now, how mathematically, what in nonlinear dynamics, nonlinear dynamic sense, or mathematically, how it means? I mean, if you really want to calculate, forget about the C1 and CRO. You just take any equation like this, any clients, and I want to check if this part is a steady state or a non-steady state. I mean, stable steady state or unstable steady state. So what you need to do, you have to take a point near this steady state. Let's say I'm just considering this steady state and now want to consider if it is a want to estimate that is a stable state or unstable steady state. So if you actually consider a point here, okay, and then so this point has some C1 values, means some X values, and in this there's a Y values, it could be 9, 10 or something values, and you put that number in the equation. And if you see that equation, in most of the cases, I mean, this is sign convention, if you just putting this value, if your equation gives a value of negative, right? If the direction, that means the equation, I mean, if you bring this equation here, actually equation, if negative means equation is trying to come towards this point. So you consider many points around this steady state, okay? Around this 183, 60 degrees or whatever. If it's a two-dimensional, it is 180. Just you actually go all these points and see that what is the direction of that particular equation at that point. If you see that all of them are negative, means they're attracting towards this steady state, this is called stable steady state. Okay. And what we'll see here, if it is unstable steady state, you will see if you take this point and you again calculate, you will see the values is positive, means because actually it's a vector space, we're talking about in terms of the vector. So you will see that its direction is, if it is positive, this is a sign convention, right? Negative and positive doesn't mean anything other than sign convention. So you can actually topple that and do it in your way. But this is the way that if it is positive, this is actually, it looks like it is repulsive, the direction. So this point's direction would be the other way. So though it is steady state, this is an unstable steady state. Okay. And in case of, so again, I'll go to the another page, so the much better, bigger picture. Okay. So if you remember the picture I have shown you, and it looks, so this is a stable steady state 1. It is unstable steady state, and this is stable steady state 2, which means if you check the points, this way it is attracting towards this point. Here they're repulsing from this point, and here also steady state. The stable steady state means it is also attracting from this. So this is a mathematical thing. Now, what is the physical meaning of this, or a chemical meaning in terms of the switch, what we are studying in the C1 and CRO lysis-lysogeny switch, and we'll see what is the means in terms of the synthetic biology. Now, so again, I come to the mathematics and then go to the physical meaning. So which basically means that if you steady state means nothing is changing at some point. Now, it is possible that you perturb the system. When you perturb the system, basically means from this point, you are perturbing the system towards here. So when you're perturbing this system, instead of going out, it will again gets attracted toward the system. That's the stable steady state. And in the unstable steady state, if it is your something is here, you just perturb a little bit, okay, it's get just repel. I mean, it got the repulsion from this, so it will go either this side or this side. Okay, so which basically means that here, if you want to perturb your system, just see in the mathematical sense. I'll come in the physical sense in a moment. So if you just perturb this system, it will always come back unless you give a very big perturbation. So perturbation is show that your point comes here, then you will see that this is now get attracted to this point. So basically, so this is the perturbation is required. Otherwise, what means that if you give a little perturbation, if you give little perturbation, it will either attract it here. If your system is present somewhere here, even if you give a little perturbation, it will go in this direction. Now, think about your light switch. So in my first class, I tried to explain that how light switch has a memory. So what I said that if you actually turn on the light switch in your home or wherever you are working now at this moment, if you turn it on, then you press your finger and then take your fingers off, still it present there. Okay. Now, if you turn it off and you pressed your finger, you take your fingers off, still it is turned off. Right? So those are basically stable steady state. Now that you know, right? Now think about do an thought experiment that you in your switch, let's say it is at this moment it is turned on. It is a one stable steady state. Now you give just a little press there. If you give a little press, okay, sometimes you will see that little press it is turned off, but if you take your fingers off, it's again on because it won't go to the other, I mean, real off state. Real off state, you know, in the switch, we have always get a sound like cut, right? So you can imagine if you slowly do that, you can imagine it will actually crossing at a certain moment, certain balance, so to give this your cut sound, right? Before that, if you don't did I mean, if you think about very slowly actually turning off or turning on that switch, if you just give a little press, your light switch maybe from on to off, but if you take your fingers off, then again it goes towards the on. So which basically means here, so mathematically, if you want to see in your switch, you have a stable steady state one, let's say it is on, you just give a little press. When you little press, it comes from here, so it is turned off. If you just now release your finger, it cannot go here because this perturbation is not good enough to cross this unstable steady state, but it will be attracted towards it and it will not. Now to come here from your stable steady state is on state to the off state of your switch, you have to give a big perturbation. And when you give the perturbation, this will sound as a cut, and this cut is going to be your turned off position. And you remember, if you do it slowly, just for experiment, you always see that it is, as you can feel that it is crossing some point. After some point, it will just coming here. And from this, when you coming goes there, from on some off to on state, you know that you have to go, you have to cross a middle state. Unless you cross the middle state, it will never come. So I mean, I imagine you can actually do that in the Throxter experiment by yourself, and you can do that experiment here. So that's the physical meaning of that. So exactly what is happening in terms of the C1 and CRO switches or any other non-dynamical system where it looks like things. So when we have a lot of C1, that your system is the lysogeny state here, right? Now, if there is a fluctuation in the C1 amount here, a little bit fluctuation, okay, so it goes from the steady state here, but it cannot go to this lytic state because it's attracted to us. So little perturbation cannot make anything. But when you have a big perturbation, like you give the UV radiation, that UV radiation is degrading your C1 protein. So it is a very big perturbation. That time, your system actually goes from here to here. It's crossed this unstable steady state and goes towards the stable steady state 1. That is the lytic phase. Okay. So this is, I mean, I try to explain in terms of physical and chemical meaning, but okay, so it works like basically that. Now again, I'll go to my slides, and we'll see different things here. So hope you can see my PowerPoint. If you cannot see, please let me know. So that's the slide I showed, that the stable steady state 1, unstable steady state 1, and stable steady state 2. So what are the other picture means? So this stable steady state or whatever, this kind of the system is only present when we have some kinetic, very kinetic parameters with this, all this. So the equation I tried to actually show in um, uh, in the paper and pencil, that is looks like that's dCRO dt is equal to function of CRO and C1. That's F1 function, and another is the F2 function. So this function definitely have some parameters. So if only have some parameters, then you only get this kind of the behavior, and that is completely evolutionary tuned, right? Now, if you change the parameters, you can actually come to the other kind of the system. So which basically means by changing the parameters, you can actually come from a switch behavior to a non-switch behavior, which basically means the other way around. So you can probably build, if you understand that particular nonlinear dynamics of the switch, okay, then probably you can build a switch. So so that's how to build. So here is just a thing what I already explained, that what the stable steady state means and what's the unstable steady state means. Stable steady state means it is attracting. Unstable steady state means it is actually repulsing from there. That's I already explained. So here this is the slide I have also shown you in my first class, because this is going to tell you how to make a biological switch. Now we have the understanding of this nonlinear dynamics, and in nonlinear dynamics, instead of the C1 and the CRO, we took a different other proteins, and that proteins is basically one, they took the laci, and another protein they took as the TETR, okay? And GFP is just connected with this R1 protein, it's TETR protein, just to see that's what is happening inside the cell, to see that instead measuring. So now we developed the similar kind of situation. As I told in the very first thing, that this is the qualitative principle. This qualitative principle of a switch you can get from the RS latch, okay? So what it means, it is a two mutually repressing promoter gene pairs. But as I said, that in order to get so for example, here, in order to get this kind, so you need this kind of behavior in order to make a switch, to build a switch. But if you just connect these two things, most of the cases you will get something like that, right? Here is the CRO, CR1, but here is that not, it is the laci and some other protein TETR. You look something like that, because most of the cases that will happen. So you have to bring its parameter, those equation, what I saw, like the F1 CRO C1 and F2 as a function of CRO C1. So there, it has also parameters, like it could be a 2x plus 3, some has a 5x plus c. Maybe these 2 and 5 numbers won't give you this kind of the switch. You have to bring probably some number. Let's say someone is a 300.2, someone is a 12.7. We do not know, but depend on the equation, that how which parameters will bring you this kind of the behavior. And here exactly what they have done, and this is, as I said, this is probably taken as one of the first example of the synthetic biology, which that is why people consider 2000 as the starting point of the synthetic biology. So you have this equation. From this equation, here is this is the parameters, that exactly at which parameter region, whatever is the alpha 1 and alpha 2 would be the parameters. So at which level, when the alpha 1, what is the interrelation between alpha 1, alpha 2, in order to get this bistable. That is, bistable means two stable states, right? Bistable behavior. So only at this parameter range you will get this bistable behavior. So when you know these parameters, interrelation between them, now you can come to your qualitative principle, and now biologically or biochemically you are changing it. What you are changing? So what to actually bring your parameters in that level, what you can change? As I said, I mean, in one of my classes, that there are some parameters when you make a synthetic genetic circuit. So we look at the four things. One is the promoter. So you can change its promoter sequence, something to change the way the transcription factor binds there, the way that RNA polymerase binds there. You can actually increase or decrease that binding, but that will change some of the parameters. Another thing I talked about is that ribosome binding sites, because ribosome binding sites is during the translation, it has to bind with the that's mRNA, has to bind with the ribosome, and it depends on the sequence of this ribosome binding site. So you can engineer there by changing its sequence in order to give a more translational rate or reduced translational rate. So you can actually develop a lot of different translation rates varying from very low to very high, right? So that is another changing area. Another changing area is the gene. I'm definitely either you want to use the LAC-I gene or you want to use the Tater gene. That is the things. And the last thing, what I said, is the transcription terminator. If you see this plasmid, this is a plasmid. Here you can see all these things. The promoter, you can see the RBS, you can see the gene, and then you can see the transcription terminator. So all are very important, right? So you can change all those things in order to bring the parameters in that the bistable region. So that is that way you can actually develop a biological switch, right? So that is the simplest biological switch you developed based on the idea of this switch from the engineering as well as the lysis and lysogeny and nonlinear dynamics, and but you haven't used any of the promoter or the genes what has been used in the lysic and lightning. So this is understanding the system by building. So when we have started understanding the switch, now it is a bistable switch. People also, like last year, actually, I'm not going to in the details of that, but people also developed a multi-stable switch. Because multi-stable switch is very, very important. So they have developed multi-stable switch in bacteria, but multi-stable switch are very, very important in the eukaryotic system. Either the developmental switch and a lot of other kind of the switch are present, gene regulation in the eukaryotic cell, which is not just the two stable states, but there's the three stable states. And based on the parameters of the perturbation, it can go from one state to another state to another. Okay, so that's biology super. So when you start building this, you can start understanding its parameters, understanding its range, and so that you can say that at which range a biological system is going to work the most. So we start understanding this kind of system, and we know that how difficult it is to make a multi-stable system than in a bistable system. So now we can understand or start understanding it, how evolution is making from a bistable system towards a multi-stable system. So what kind of the parameters we had to use, so what kind of the gene networks had to be used, and so on, so on. So this is an exciting time because now you can actually start understanding things by building. So this is one example of the building. I'll just work on this switch. So another part of the synthetic biology where you can actually control a switch. Okay, so in this work, what happened? So you have this lysis and lysogeny switch. So here, the scientists actually developed a system which can control that switch. What it means? So their idea is actually put some synthetic genetic circuits inside the bacteria in a lysogeny state of this bacteria, so that particular system is stay dormant. And when there is an UV, you give a perturbation to the system, the UV, so which basically means C1 is getting reduced. Now that biochemistry change should be sensed by the synthetic gene circuits, and it start making more and more C1, so that it only stays at this the stable steady state 1 or 2, whatever I'm saying with the for the lysogeny. So it won't never go to that system. So how if people actually develop that? So they think about in terms of the control engineering or the control theory. So if any of you have the engineering background, you know that's how a control systems looks like. But very simply to tell you that you have a system, and this system has steady production of a C1, right? And then you have another loop, which is the CRO, which has to be kept low. So that is, let's say, it could be a chemical reactor, it could be a mechanical device where you at a particular specific time, you want a particular system to be work on, and others should be stopped. It never should turn go from here to here. But there is always a disturbance in the system, and this here is the disturbance is coming from the UV radiation. If you give the UV radiation, and when you give this UV radiation, it gives a cleavage to this C1 concentration, and that means the C1 is get reduced. Now you developed some synthetic controller based on these engineered promoters and gene in such a way they can sense that reduction in the C1 protein. If they sense this C1 protein reduction, then it starts producing the C1, okay, so to make this C1 always as high as the system. Now it is important that it is making the C1 is not going to stop the system from going from this system to there, because it has to make the C1 fast enough that before this system, I mean, if once this system go to the CRO, we cannot do anything. So, but this controller has, so that you have to take lot of kinetic parameters into account, because the C1 has to produce fast enough so that before going to the CRO, C1 concentration comes back. Now we think about that is exactly happens in your fridge. So if you have some old fridge or refrigerator in your home, sometimes you'll see there is a gong gong sound. That sound is actually coming from the compressor. So what is happening? Let's say you start. So let's say the 25 degrees the ambient temperature. Now you want to bring it down to the 4 degrees. You start your refrigerator, and then compressor start working. But compressor doesn't know where to stop. So it is coming down, coming down, coming down, coming down, coming down. So you have a controller, a sensor, and a temperature sensor and a controller. When the temperature sensor set, I mean, if you set your fridge in the 4 degrees centigrade, and if it is come down a little bit from the 4 degrees centigrade, what happens? That compressor actually stops. Only the compressor stops, then again the temperature starts going to rise. And when the temperature going to rise, then again the controller actually sense there's a change in the temperature, and again that compressor starts. That is another go-go sound that's going to start here. So now it is important that it has to work fast. Means it is not possible that like your temperature shoots up from 4 degree to 25 degree again, and it takes like 1 month to come down in the 4 degree. No, you have to it actually bring down to the 4 degree few minutes. Okay, so this dynamics here is also important. So that is why the engineering principles you are using here in the synthetic biology, you make your promoter gene, your circuit or synthetic controller in such a way it is not only actually sense and produce the C1, but it has to do is the fast enough, right? So that's the so this way you can actually control the switch. Now, here is some of the experimental results, what you can see. So if you see, if you give, so there's a K12 strain of the E. coli. Now, that is basically a lysogenic strain. And if you give the UV, when you do not have a controller, you can see the cells are started growing, and then it is coming, the cells, the bacteria cells are dying, because this is the, you're measuring OD, and this is the time after the UV induction. But if you don't give any UV, the cells are just going. Now, if you put those controllers, there are several controllers they made, and if you put those controllers inside the cell, and now if you give the UV radiation, you can see they are not dying like this, but they're going. So this means that these controllers are basically working. But it is not only working, but it just shows that it actually automatically detects it. So here you can see that if you take those kind of the cells and divide them, I mean, where you have this controller, and then give the UV, then only you can. So because what I have talked about in this system, they have some, I mean, they definitely have the C1, but they also have a green fluorescence protein, because you can measure that, you can see that, which is actually proportional to the C1 protein you're going to produce. So here what you see, that after some time, after you give the UV, your GFP expression increasing, right? GFP expression increasing and increasing, and if there's no UV, no GFP expression is increasing. So which basically says that it automatically can detect only. That is why after detecting, it is, I mean, after the UV radiation, then it starts increasing this, the GFP or the C1. That's the proof of that particular thing. So it has another property because you can turn off. Like if you think about this is software, like an antivirus software for the bacteria, which actually, which allow you to actually stop the virus attack in the lysis and lysogeny, which means that as it's a software, you have the switch to turn off that software from the outside. So that is how we can do that. They actually used a C1 protein, a temperature sensitive C1 protein. So if you do that whole experiment in 32 degrees centigrade, this will work as your controller system, a proper controller system. But if you want to turn it off, you have to actually increase the temperature to the 42 degrees centigrade. In this 42 degrees centigrade, that temperature sensitive C1 actually dies off, and your system, you can actually turn off the system. So now you can see the cells are dying. So you have the system, a lot of dynamics. You can actually control these things. This experiment shows that it is not just increasing the C1, but it sets off when this everything goes things and sets off, and that is what you can see, that how EJP is increasing, and after some time EJP is coming exactly in the basal level state. So this says that this working exactly like a device. Now, what in my first or second class I couldn't remember, when I talk about how do the mathematical modeling, I showed much simpler modeling, but when at the very beginning, when people actually tried to this kind of the synthetic biology, they did a different kind of the modeling. So here they write all the equations, means all the chemicals, whatever the chemical things are happening. So here you can see what is the equilibrium reactions, what is the transcription and translation reaction, what is the transcription translation reaction of something, translation, okay, a lot of different translation degradation reaction, which means you need, for each of them, you need some of the parameters, somehow you need the parameter values. Either you can find those values or you have to actually measure those values, right? For C1 and CRO, it is always present there. So you can see there's a lot of equations here. So and you write differential equation for each of them, and then it looks something like that, very cumbersome. So there's actually I can see here at least 25 different differential equation, and you have to solve it simultaneously. So you solve in the MATLAB with a new, I mean, there's no analytical solution possible, so you solve this numerically, and then here it shows that what is the connection between the experimental prediction and experimental results, and you will see in the most of the cases this experimental result and mathematical prediction matches, because you see if these two colors, I mean two bars, if these two colors from the top and colors from the bottom match, means that experimental and things match. Wherever it did not match, from here you can see that experimental result and the prediction didn't match, but most of the cases is matched. So it is a very good thing. So you do have a molecular modeling kind of differential equation kind of modeling. You are developing a biological system, and actually in a population level you are measuring. So this is in that way it's a good mathematical modeling. But as I said, at this moment people generally don't do those kind of differential equation. They use Hill function kind of the equation, but you can do any of them. So this is a thing. So here they have a lot of different kind of the controllers, and they try to say which have the which kind of the property. Probably only one of them has a very good property for all of them. Others have if you think about like a software, then you can think about, okay, this is the best version of the software, this is the student version of the software, where all the properties are not there, something like that. So till now what I tried to tell you, that's how so the idea that using synthetic biology, you can build things. That's in here I give the example of the switch. So you can build the switch, how we are going to build the switch, and using the similar kind of principle, you can, that means you can also control that switch. So that is the second example I have given to you. Now I'll just switch the gear from here, and I'll go basically in the space bioengineering problems. So as I explained in my first class also, there's that today's our human space travel is different. That goal of the human space travel is different than what was 50 years back. Now you want to go, now people, you can see that how inborn exploration, how this is the cartoon plan, how you want to actually go to the Mars. So because you have to go through a lot of obstacles, a lot of the plans, and this is kind of the plans how can actually go there. So from this Earth to coming to the here, it will take months. Even if you go is the fastest way, it will take several months to come to the Mars, and again from the Mars you have to go to the Earth. It will take another several months, and now you want to stay on the Mars for a month or so, right? So it will be like in one way it is like I don't know, maybe 7-8 months to more than a year. So if one astronaut has to come there, and so there's a lot of this thing. So what are the obstacles here, and why do you need space, and why want to take so much pain in going to the space? So there is a lot of reason, right? So space, I mean, if you have seen the Star Trek movie, they always tell the space is the ultimate frontier. So this is space, I mean, from our childhood, whoever loves science, I mean, all of you definitely, probably you were always astonished by looking at the sky, that how those stars are coming. Okay, and if you look at the whole, there's the sky, in the night sky, there's so many stars there, several billions years old, and they're coming from several light years distance, and what is our position in this universe? So those are kind of a question, in scientific as well as the philosophical question comes when we actually look at the space. So the space always takes a very special space in our heart, right? So that's why one of the reason that's why we want to explore the space again and again, and as long as we actually send the human to the Mars, that would be another milestone for the human search for the space or other things, right? So that is definitely one of the reasons. But apart from that, there are some practical usefulness that we can also think about for the space. So one of them, I mean, okay, so this is the same thing I tried to tell that, yeah, you are going to the Mars and you want to make a research base and something. So apart from that, what there is a one thing I can think that we are talking about the semiconductor engineering or semiconductor processing in the space. So if you think about the semiconductor, so basically like the Intel chips in your computers. So these chips are made generally from a silicon wafer, right? Now you have to print on this silicon wafer. So there is a two important thing. I mean, there is a two obstacles to make there. One thing is that when you actually print nowadays the chips, I mean, you can actually print a lot of circuits in a very small amount of the chip, right? So what, if I remember correctly, so at this moment, if you think about in terms of wear, there's no wear in that way, but in the silicon chip. But if you think about in terms of the wear, so the two circuit, the distance between the two circuits or the two wear, okay, at this moment you can actually bring down to somewhere around the 20 or 25 nanometer range. Okay, so it's completely small. That means in the 25 nanometers difference, you can actually draw two different circuits. Okay, before it was like micrometers, before centimeters, and so on, so on. So now we are coming. That is why you can see in your mobile have a lot of memory, lot of or even a computational power, because you can actually bring that difference between these two wears. I mean, wears is a wrong term, but if you actually this printing wears, this comes to in the nanometer level. Now, this gives a very problematic situation because you need a cold room, a clean room. Why is that so? Because if there is a particle, so whenever you are actually doing something, there is a lot of particles are going around, right? Lot of dust particles, and those dust particles, if any of them is more than 21 nanometer, 25 nanometer, that is the case, right? So there's a lot of things in the several nanometers, 1,000, even in the micrometer region. So if that actually sits on your chip, then your circuits has been gone, right? Because there's only 21 and you don't want a short circuit or a very particular particles to come there, right? So you need a clean room, and this clean room looks like here, like in biological phase 4 system. Here you can see the engineers are wearing the dress like the COVID warriors, right? Why there? But not to for the contamination to the human, but they don't want any contamination for the human to go to the semiconductor. So making this kind of a clean room and maintaining it, because whatever the particles are coming to the room, you have to always pump it out. So it takes a huge amount of cost, energy cost, and the money, right? So that is one obstacles. And another obstacle is the silicon itself is so fragile that it actually breaks down due to its own weight, which basically means you cannot make a big silicon wafer. Okay, so you cannot make a big silicon wafer, because if you make a big silicon wafer, it will just breaks down by itself due to the gravity. So you have to make a small silicon wafer to make this big. Now, space can give a solution to both of the problem. First of all, space does not have a gravity, so we can make a lot bigger silicon wafer, right? Because there is no gravity, it won't pull it down the silicon, and it won't break down by itself. So you can, first thing, in the space there is no gravity, so you can make a big silicon wafer. Second, space doesn't have any dust particles, so you do not have to maintain this kind of the very costly clean rooms. And people actually do, so there's a lot of I mean, kind of economics work. People actually try to do the cost benefit analysis of the semiconductor processing on the space. And people also found that in many cases, if you actually process the semiconductor on the space, then it takes from 10 to 20% less cost than what you want to make in the art. Okay, so imagine that if you think about a semiconductor industry in the space, that could be even important, because you can actually increase the area of your silicon wafer. You can make bigger silicon wafer. You don't need the clean room, and it could be a little cheaper. So that is one of the possibility. People think the space can be used um, for human good. And another one, that, as I said, that when I talked about just a tissue engineering, when this tissue engineering, the way we do in the art, we have a material scaffold, make a three-dimensional thing, and then you embed your cell, and that cell actually grow towards that three-dimensional, right? But the space doesn't have the gravity, so if you just grow the cell in the space, okay, it will form a three-dimensional structure. Here are the two examples. One is one artificially produced colon cancer tumor in the ISS and the left hand side. But if you grow those kind of just colon cancer cell given in a dish and put it in the lab, the way all the biologists do in the lab, you won't see this kind of the three-dimensional. This is like a seat like the structure on the plates you will get, right? And another is that right hand side, you can show there's a 3D liver tissue growth in the face. So you don't do any material scaffold, don't give anything, it will just actually grow like a three-dimensional. So it shows that if the tissue engineering may have some application in the space, because it has a very low gravity, or it doesn't have a gravity. So that's so this is another area of these things where the space can be useful in near future for, again, human good, right? So one important part, then it is coming the gravity in the space, which called is the microgravity, right? And this how this microgravity will work on a spaceship, that is not, I mean, before the scientists actually thought about it, the people in the literature thought about it. And one is from the Jules Verne's Earth to Moon, and it is published in 1865, where he actually imagined. So this picture you can see here in the left hand side. So they're going towards from Earth to moon and the space. At a certain point, they actually found there is no gravity, and they start actually floating. And this is, that time nobody know about this space science, right? 1865. So how imaginative how those people are, like Jules Verne, you can see here. And this is 1953. Probably you have studied, I mean, you read actually the Tintins. I love the Tintin very much. And this is the explorers on the moon. You can see the whiskey from the Captain Haddock is actually, they started floating, and the whiskey looks like a sphere. And that is exactly happens when the cell grows, because there's no gravity. That is why they make this three-dimensional. So it is a literature, the effect of the microgravity we know from a long time. But actually for the astronauts, this microgravity is not a good thing. So the space is basically a health hazard for the astronaut. So there's a two region. One is definitely the radiation. I'm not going to talk about the radiation. I'm just talking about the microgravity. So due to the microgravity, there's many things happens. So one thing is like your skeletal, there's a lot of bone loss happens. Okay, and gives osteoporosis, bone loss, so many calcium and sodium metabolism dysregulation. Okay, and then muscle loads, because therefore the muscle degradation happens. Okay, motion sickness in the space. There is increased protein turnover, and because the whole body has the fluid, right? We have the blood, and blood goes the way we are designed, because we are not designed for the space, we designed for the earth, for the gravity, right? And that's why the whole evolution happens. So if you go, so there is always an gravitational pull on our bodily system, that the blood is coming where, that is why heart get to pump these things, right? So now if you go to the space, there is no gravity, so there is a lot of fluid accumulation happens at the many spaces in our body, and that makes the health hazard for the astronauts. But what is very interesting is, one happens in the microgravity, there is the immune response actually got reduced, right? What are the other parts we can actually explain in terms of the mechanics, either the bone loss, or the muscle loss, or the fluid problem, or the blood flow problem in the space, but very difficult to explain the reduced immune response, because this is exactly a cellular event, okay? And how a cellular event is happening, the genetic effect is happening in the space, this is always a mystery. So this is not the only thing. So this is something you have to actually look at more biology than the physics or the mechanics, because it is happening exactly in the genetic level and the molecular level in the cell. So the reduced immune performance. At the very same time, there is something happens to the bacteria. So like salmonella, those kind of bacteria got virus. So we have in our gut, we have salmonella at this moment, but it is not doing any harm. So, but if you go to the space, that salmonella got virulent and will infect you. And these people know from this, starting from the Apollo mission, because they got some urine infection due to virulence. At the very same time, people actually saw they got also the drug resistance. So if you the same bacteria, if you send to the space, that bacteria in the space, they'll give you a, they're more resistance to the drug than on the Earth. So these two things actually a clear combination, right, for the astronauts, and also because the bacteria is so tiny mass it has, so we really don't know that how this the change of the gravity is actually affecting that. Though there is one theory, so the theory is like, so if you okay, so first of all, let's talk. Let me talk about the biology. So there is a lot of biological changes are happening. So means a lot of gene expression changes are happening, and the way people studied that. So you take the human cell or the bacterial cell, or you send to the space in the International Space Station, or there's some microgravity simulator are available on the Earth, and you can actually run those experiments on the Earth, and then you do the transcriptomics and proteomics, and then you try to understand what is the genome wide gene expression on the protein expression, and then you start to understand what is the pathway is changing due to microgravity. That is very good, but this does not answer that how that tiny change in the gravity for a tiny tiny particle like a bacteria, how that little change in the force is changing everything in the genetic and proteomics level. So one explanation, though there is no proof, the explanation is in the bacteria, what happens, so there's a fluid around the bacteria, and this fluid, when it go to the space, it gives a low shear stress. Its shear stress actually get reduced. Okay, that's known. That is the fluid dynamics of the fluid dynamics in the space in the microgravity. And when it happens, then the exchange of the transport phenomena of vehicle metabolites, food, and everything got hampered, because if there's a turbulence around the bacteria, then there is always a good exchange between the foods particles, the toxins, and so on. But when there is a low shear stress, it is only just static fluid layer, then the exchange of the materials between the bacteria and its environment is got hampered. That's probably is going to give this kind of the big change in the gene expression and phenotypic change. As I said, it's got virulent, it got drug resistance, acid resistance, and so on, so on. And we actually going to study those kind of the space thing like that. But we cannot send every day to the International Space Station. So how we are going to study in our lab? So on the Earth, the way we actually study, there is a lot of microgravity simulating instruments are available. One is called clinostat, or here is one particular type of the clinostat I showed. It's called RWV, or the rotating wall vessel. So this is designed by the NASA. What they have, so this is a reactor. This is a kind of a disk reactor. What happens? It rotates in this way, and you fill it with your bacteria or the cells or mammalian cells or whatever with that. Now, when it rotates, and your gravity vector is in this way, so if it won't rotate, so if there is a particle here, due to the gravity, it will settle down here. But when it rotates, its axis is always changing. So means before that, it cannot come down. Before it is coming down, it will go. This part will go on the top, right? Before it is coming down, it will go in the top, which basically means it will stay somewhere in the middle, in the suspended way. And that is the effect of the microgravity you can actually generate here. So here is another. So in order to control, if you just flip it, then your gravity vector and your rotation vector, I mean, if it is in the same work in this way, this kind of way, then this gravity vector is not getting cancelled. So in this way, you can actually generate a microgravity. Similarly, there is another machine is called the random positioning machine. It is better than the RWV, because here, this machine, you can actually put your, see, there are some, here you can see, this is the plates where, or the tray where, or the flask where you have the cells. And now, instead of rotating in one direction, it rotates randomly throughout this 360. So it is cancelling the gravity vector from a 360-degree angle. So that's why it looks a little better than the RWV, because RWV is only the one thing. Here you can also calculate what kind of the microgravity you are generating through that. So this is another way you can actually use in your lab to generate the microgravity, put your cell, and then analyze. So another kind of things people used, and this is called the magnetic levitation. So what is the magnetic levitation? With the magnet, you can actually put an object, okay, just floating against the gravity. But when it comes to a biological material, okay, because this material there could be a magnet, any magnet you probably use, but when it comes to the biological material, a frog is not a magnet in one sense, right? It is not like a magnet, what we know as a magnet. But if you want to make a magnetic field such a way that this frog can be floated, you need a very high magnetic field, and you do use by superconductor magnets, right? And what it takes, like people actually shows the insect, frog, and mouse can be levitated like using this magnetic. And so what is the magnetism in this frog or the mouse or insect? Because there's a lot of water. The body's 70% is the water, and this water is the diamagnetics. Okay, so when you think about the magnet actually attracts an iron is a ferromagnet, right? And the diamagnet, the water is the diamagnet. I mean, just you put the water and just put a magnet, that water won't come here. So you need a very high level of the magnetic force. That is exactly happening here, so that the whole animal we actually go to the floor. So this is the microgravity simulating things. So another people can do that, and it is called acoustic levitation. So instead of the magnetic force, now you give some sound force, very high intensity sound. So the sound, you know that, but definitely you must have air here, and because it gives a static wave there, and the static wave adds this force to keep the particles there. And you can see here on one experiment, due to using this acoustic levitation, it is an insect. It looks like an ant. It is actually floating in between against the gravity. Okay, so now, however, the one thing is important, that when you actually try to understand the change of the biology due to the microgravity, if you use this magnetic levitation or the acoustic levitation, but it can change some of the other property of the biological system, right? So it's not always advisable to use, when you study the biological materials, to use this kind of the system, because we don't know what is the effect of the magnetism and how much of the effect of the microgravity is coming. So, but in general way, those RWV and RPM is much better things, but those are mainly for, you can understand, mostly those are for the cells, right? So those are the things. So here, one example, what we try to show that people actually developed a microgravity responsive or microgravity sensing synthetic genetic circuit. So how they have developed? So there is a HFQ protein. That HFQ protein gets downregulated in microgravity. So now, if you can develop a genetic circuit which dependent on the HFQ system, and that really happens because in a translational inhibition from a if you have some antisense RNA, okay, against something, this HFQ is required for this antisensing property. Now in the microgravity, this HFQ it's get reduced, which means that antisensing capacity of the cell get reduced. So when you use this and that principle and develop a synthetic genetic circuit to do that, and do the experiment, actually you can see this works as a microgravity sensor. It is in the microgravity it looks like something, and one gravity it is look like something. This is the control experiment. So this is the microgravity sensor you can develop in the bacteria, and you can use that sensor to connect some other biological process to the microgravity. Now you use the microgravity as a knob or a stimulus, a physical stimulus for the system. How? Here they showed that there is a one protein called FTAZ. This protein is required for the cell division. So if there is a problem in this FTAZ thing, then what you will get? You will get this cell won't divide, but it will just elongate it. So you'll get an elongated kind of things. Now they use their synthetic genetic circuits connecting with the antisense RNA against this FTSG. So what you see in the Earth's gravity, you can see these elongated cells. This is E. coli, elongated E. coli. Now when you bring it back to the microgravity, what will happen? This HFQ level will basically decrease, and means that this antisense work of this RNA against this FTSG won't be good. That's why this elongated things comes to the normal size. They started dividing again. So this is one good example of where you can develop a synthetic gene circuit, connect with the microgravity to do a lot of work inside the cell. So now, if you go to the NASA website, then you will see they have a bioengineering branch. This bioengineering branch is not that old. This is a new, because now we know that in the space that the bioengineering actually take a lot of it can solve a lot of problem. So they actually divide these things in. So what are their immediate goal is that atmosphere means not the climate. Atmosphere means that international space and atmosphere under the International Space Station inside. So atmosphere's revitalization that can be done with biotechnology or bioengineering. Water recovery, that is very important, because we are always losing water there. So we need whatever the is coming as an effluent or as a waste, that water has to recover in the proper water. Waste management, another important thing. If you want to stay in the ISS, they use synthetic biology. I mean, they put it synthetic biology as other, but it connects to everything like critical sources for exploration, food, life support, and materials. Means it's connected with the water recovery, waste management. Also the biofuels, systems level engineering, because the whole architecture analysis, model and simulation of this, whatever the life support system you are developing in the biology, a similar kind of the mechanical engineering, electrical engineering kind of the system engineering is required. And definitely the lot of analytical chemistry things are required there. So you can, so here there's some area what NASA identified at the immediate area of using the bioengineering here. And if you go, there's a lot of, I just showed that some of the publication from 2020 from the NASA, that what kind of things they are doing. So here you see the crew member microbiome. So they're trying to understand how it is working the gravity, microgravity. So NASA Gene Lab interfaces for omics data, visualization omic data, validation of new rodent experimental system investigate. Okay, space flight and simulated microgravity condition increase virulence in some some pathogen in Drosophila. So so there's a lot of this kind of the studies are at this moment going on. And here is something like space radiation. So how NASA Gene Lab platform utilized for biological response to the space radiation in the animal model. So these are the kind of the things people are using. So this is the experiment. This is the example I showed, that what people are also thinking, using synthetic biology to make a self-replicating habitat, especially for the Mars. Okay, and there's started working on it. I talked about these reactors where you can use the basically the chlorophyll, I mean the cyanobacteria or algae to develop a lot of food and chemicals, taking the sunlight. And here is the synthetic metabolic engineering are important. So this is a reactor design only. So this is another project from the Europe, and this is the it's called MELISA project. And here the whole idea, a closed loop life support system or ecology you want to make inside the International Space Station. So wherever, so the way on the Earth we have the biogeochemical cycles, like the carbon cycles, the water cycles, the nitrogen cycles. You want to develop a closed looped system in the ISS, which has the all kinds of stuff. I mean, like the Earth, it must have all kinds of different cycles you can control there. So that's called the MELISA project. People are working for a long time on that. This is a very big engineering project, and definitely people are using synthetic biology there. And here, this one I also showed that this is the area people thought where synthetic biology can give some boost in the space bioengineering, starting from resource utilization, manufacturing, life support, space medicine, space cybernetics, and terraforming. So when you talk about the terraforming at this moment, nobody is thinking to change the Earth's atmosphere like the Mars atmosphere just now. But they think about in terms of the para-terraforming. What is para-terraforming? Let's say you get, so if there is a mountain on the Mars and you get a cave, right? So the cave has every other parts are closed, only your front part is not closed. So you close the front part. So now you have a closed environment, and that closed environment we actually make the oxygen, you make the atmosphere like that. It's a small area, but you can do that. So like the MELISA project and everything can do that. So the people, when we talk terraforming at very this moment, what people, the synthetic biologists think about this para-terraforming, not the whole atmosphere just at this moment, but just to mention. So these are the area where synthetic biologists are going. So I'm coming at the last. Actually, here I just show just 2 slides. So one slide is if you want to go for a further reading on the synthetic biology. These are random in one sense, but if you just study few of them, you can actually get the idea and you know that from where you have to go. But I also talk about some books. This says that how in a synthetic biology things, how you're going to do the model, I mean, in terms of the mathematics, what kind of those things. And another book is the tools and applications in the synthetic biology. These are the 2 books. Others are different kind of the papers. But the papers which I actually discussed throughout our things, I haven't put there, but these are some of the other things where you can actually see how we actually develop synthetic genetic circuits. And another set of further reading, if you are interested in the space bioengineering. So there are 2 books I'd suggest. And so definitely the space bioengineering is very new and people do not know what to do. I mean, because it is not just the bioengineering, but the space bioengineering for the space. So you won't get, there's a lot of space biology books and papers you'll get, but not many for the space bioengineering. But still, what a very basic book. One is about the space and life. It is talk about the space biology by Hubert Planel. Another book is the astrobiology by Charles Cockle. Those are the 2 very important books if you are interested to things. And then a couple of papers. And another is definitely the NASA bioengineering website, which will give you a lot of idea that what they're doing, what they want to do. So with that, I'd like to stop here. And I thank all of you for this synthetic biology. I also, I realize that there's many students are there. What I tried to give you just, it is not a full synthetic biology course, it can't be, but I just tried to give you some idea that how to develop synthetic genetic circuits and what could be its application. So you can see what I try to, the message, the take home message, at least what I actually tried to communicate with you, that the possibilities are enormous. It is just your imagination, which whatever you can imagine in using biology to do, the synthetic biology can help you to do that. So we really do not know what are the rules, all the rules at this moment probably. So if you are interested in the research, biology research, to take the research as a career, I'll request you to give a thought about synthetic biology any field of the synthetic biology, and it has an unprecedented power that what we believe, and you can do anything. Many people think the synthetic biology at this moment is exactly at the same thing, the way the computers actually developed that time. So at the very beginning, nobody knows what to do with the computers, but now we know what the computer revolution is. So the synthetic biology is exactly at that phase of this computer generation. So think about it, because synthetic biology also. So when you, I may not be correct, but many people actually told me, or many scientists, actually my professors. So you choose a research field which will actually pick, because you are the students. I'm talking about the students. Okay, so which will pick after 20 years. That means if you start your research from now, when you will be an expert in the 20s, when the field is actually in the peak, so you will be in a very big, very good position in terms of that. So this, because one thing is, the new thing is also very exciting. At the very beginning, there's a lot of challenges, definitely a lot of frustrations, true, but lots of excitement. I think uh, uh, if you think, what I'm going to tell you again, that please just give a thought about the synthetic biology. You don't have to do synthetic biology, but before you choose, I'm just requesting you to give a thought about the synthetic biology. So I stop here. If you have any question, please let me know. Okay, so it seems there is no question from you, but if you go to my site, you definitely get my email. Probably you have. So if you have any question regarding synthetic biology, so you can always email me, not just for the course, but anything about the synthetic biology. I'll always going to help you. Thank you very much.
No questions, so we'll end.
Okay, we'll end. Thank you.